Professor Jung from UC San Diego is going to focus on more environments, naturalistic environments and interaction with task forces. Thank you.
Okay, is that on?
Yep.
Okay. First of all, I want to thank for the invitation from the organizing committee. I really appreciate this opportunity to share with you my recent studies regarding the neuro and physiological correlates of task performance in naturalistic environments. First of all, just like John, and my talk is mainly focused on normal human subject and not patients. And second of all, usually I actually run out of time, to finish my last slides, so I didn't get a chance to acknowledge my students, postdocs. So I will do that first. And this is all pictures of the lab at SWAT Center for Computational Neuroscience, which is upstairs. And we're going to do a lot of demos, live demos, tomorrow. And those people, particularly, I want to point out the people who actually develop MATLAB Toolbox has been downloaded by many, many people in the field of EEG and electrophysiological studies. And also my colleagues at National Jiao Tong University, particularly Brain Research Center, and I'm also an adjunct professor over there. So I want to acknowledge them first. The next slide shows the outline of my talk. And the first and the second part are going to focus, is that right? I'm going to focus on the human performance. Particularly, I want to sort of narrow down what I'm going to focus on. I'm going to focus on the effects of cognitive state changes on the test performance of normal subjects. And the last part of my talk, I'm going to show you some of the recent study, and that is the EEG spectrum can predict the gaming performance and signal-to-noise ratio of steady-state visual evoked potentials in single trials. Okay, the first part, I'm talking about the cognitive state and changes. Particularly, I'm interested in the lapse of attentions and drowsiness. The reason being that, as you know, the lapse of attention or drowsiness can lead to catastrophic incidents for many workers in many occupations. For instance, truck drivers. If you fall into sleep on the road, you could actually lead to car accidents. In fact, I want to point out National Sleep Foundation, or short for NSF, actually report that 60% of adult drivers have driven a vehicle while feeling drowsy, and 37% of them actually falling asleep. And this basically tells us like 40% of the people are actually lying. So because most of the times we, if particularly long-haul driving, we tend to have waves of drowsiness during driving. So we're trying to combat that. So that's what I'm focusing on. And I wish I could actually put our subjects on the road while driving to monitor their neuro and physiological activities. But it's very difficult, if ever possible, to put the drowsy subjects on the road, because that actually will put them in danger. So we end up with building a very realistic environment. And in this case, we have a real car mounted on 6 degrees of feed and motion platforms. And we have a 360 degrees immersive virtual environment. So this is a car in the middle of our VR driving simulators. And the subjects actually is riding the car, feel all these motion cues, acceleration, decelerations, and movements. We have collect data from over 500 subjects across different studies, even including the motion sickness. For instance, being the driver versus being the passenger, how your brain responds to these motions cues. So the experiment where subjects to perform is actually sustained attention test. We ask them to respond to lane deviation to the left or to the right as soon as possible with the steering wheels. So for instance, this is the case, the car cruising on the highway, and to drift to the left, you have to compensate and steering the car back to the center of the land. And very often, actually it's almost over 70% of time, we see subjects have wave of drowsiness, which is objectively measured by their reaction time to the land deviation. Okay, you can see that this particular subject, the first few minutes, perfect. And you can see the waves of drowsiness, sometimes even did not respond to land deviation for 15 seconds. These guys will be dead if we put them put him on the road. So that's the reason we want to build a naturalistic and safe environment. And one of the reasons is we want to test, or want to see if the laboratory-oriented research can be translated to real-world environments. But this is like the middle ground. And frankly speaking, I feel more comfortable to put people in the simulator as opposed to on the road. So this is a typical performance, and over 90 actually minutes. And you can see waves of drowsiness. Sometimes woke up, perform really nicely. This is reaction time. And then fight with drowsiness again. Okay, as you can expect, and I use independent component analysis, trying to analyze data, trying to separate those independent sources from the mixtures of recording, mixture of signals recorded from the scalp. Okay, we have multiple channels, and we use independent components, which was the technology I introduced to the field of biomedical signal processing in 1995. So then we can separate into independent components. Each has a time course of activation. In this case, you can see it's off, on, off, on. And with scalp topographies representing what kind of activity you will see on your scalp sensors. So that's the technology we have been using. And over the years, we actually put all those tools together and build up a graph user interface and release to the internet. So this is, Schwartz Center is the home of EEG lab. And I particularly want to acknowledge Arnold DeLong, who actually mainly put these things together and release. He's actually in the audience, as I recall. You can raise your hand if possible. And it has been downloaded by 250,000 different groups and individuals. And also another toolbox I'm going to mention is Source Information Flow Toolbox, and developed by Tim Mullen. He's also in the audience, and he is going to do a live demo tomorrow. Okay, so this approach is trying to look at network connectivity as opposed to regions of interest, how the EEG activity changes at different locations. Instead, this is trying to see the interactions among different regions. Oh, Tim is over there. And particularly, which part of the brain influences the other part of the brain. Okay, so I'm going to use this tool, and then this is for a summary slide. We look at component clusters. So basically, we got independent components for each session. Okay, each individual recording, and we separate into different components. And then we can do the clustering analysis across many, many subjects, many, many sessions. So this is the mean cluster map, and showing the bilateral occipital activities from Borman areas 18, 19. And this is almost identical components when we turn the motion platform on versus turn it off. So this is mimic the stationary conditions like you can do in the regular EEG laboratory. This is, we use motion platform, so subjects will receive not only the visual, but also the motion cues. So then we have more naturalistic input. And then you can see, I'm going to navigate you through this. This is sorted according to mean reaction time. So this is the subject response really quickly. And this is drowsiness, and subjects didn't respond until 3 seconds later after the land deviation. Okay, so you can see from the transition from awake and to drowsiness. Okay, and then this is motionless compared to motions. You can see in general, the low frequency EEG spectrum power actually increase when you have really poor performance. Okay, roughly the same. But if you look at carefully across why is jumping so fast? Across the different frequency band. So this is delta band all the way to beta band from 13 to 20 hertz. And again, this is sorted according to subject response. And you can see the monotonic increase in delta band in both conditions. Motion platform is on versus off, except the alpha power spectrum. Well, unfortunately, the alpha power spectrum has been used as a primary index of drowsiness in a laboratory setting for years. But you can see that when we have subject has motions cues input from the motion platforms, and it's not statistically significant from the baseline at all. So it's not a very reliable index. Okay, so this basically tells you in general what kind of EEG activity you expect from the transition, in the transition from awakeness to drowsiness. And also show you the differences between so-called realistic or real-world environments versus laboratory settings. And we also use SIFT, Tim Mullen's SIFT, to look at network dynamics. Particularly, this is motion platform on. This is motion platform off. You can see the activities or interactions among different regions of interest. In this case, to summarize, and when you turn on the motion platforms, you will see the major hub of your network dynamics actually is at posterior cingulate cortex versus mid-cingulate cortex when you turn it off, so sort of sensory deprived conditions. And then we look at the behavior changes. So particularly, you can see that if we have sensory deprived conditions, which means we turn the motion platform off, okay, you can see if you look at just 1 second before the length deviation, if that's dominated by MCC versus by PCC activations, you can see this is 400 milliseconds longer, which means if you look at the EEG activity right before the stimulus onset, the length deviation onset, and if that's actually dominated by PCC, it will take, on average, 400 milliseconds longer to respond to length deviations, to stimulus onset. Okay, and we also, I just told you that when we talk about the physiological changes, you can see this, we sort all the data, and we measure the dismal and also gradient between the dismal and the core body temperatures. So this is actually very tiny little changes. You can see that when reaction time increase, there's an increase in dismal temperatures and also the gradient. So you can look at, on the sort of growth scale, your body temperature changes when you get from awakeness to drowsiness. Okay, in fact, over the times, we have also, where in the engineering head, we have developed sort of brain decoding mechanism, trying to continuously and monitors your drowsiness level, detect fatigue, and we can mitigate that. In this case, usually we actually ask subjects to come in to do the pilot session. We collect data from 90 minutes of experiment, get training data, and we build a model, and you can use LDA, or you can use SVM, you can use many different classifiers. And then you can do that online, okay, to continuously monitor your brain activity to identify the changes in your cognitive state. And when the subject is drowsy, we actually send in alarm signals to alert the subject. Okay, we have published and over probably 30 journals. This is some of them actually dealing with more cognitive states monitoring. And so looks very nice, and we also very promising. We also actually implement on the cell phone, smartphone. So computational load is very minimal. You can do that on the cell phone. It seems very, very positive. But then we actually found out some practical issues. I'm going to show you. Oh, by the way, I should mention this first. So I also want to validate the use of EEG-based approach. So why you want to use EEG-based approach? If you can give the alarm feedback at the random time, that will actually alert subjects anyway, right? So you should give random time. And this is what the validation study we did. So you can see that if we give random time, sometimes we just miss and proper times to deliver the arousing feedback, you get a really long reaction time, which means you put the subject in danger. And the red trace shows the reaction time, the behavior reaction time across 45 or 60 minutes. You can see it's never go beyond 3 times of the normalized reaction time during the awakeness. So this is, as I mentioned, we're trying very hard to develop this system to also validate that, and over many, many subjects. But then we realized one problem, which is I'm going to focus on today. Mary actually mentioned this a little bit, human variability. Both across different subjects and also intra-subjects, within subject over time. Okay, I'm going to show you one slide, summarize this result. So this is called spectral correlations. Basically, for each frequency band, the EEG power, we plot the correlations between that particular frequency and test performance. So in this case, for instance, the positive correlation means when subject has performance, the power increase, okay? And you can see that across different subjects, each subject performed 2 sessions. And for subject A and subject C, they're almost identical. But subject B, you can see from session 1 to session 2 is different, okay? And also if you compare the spectral correlations from subject A and subject C, they're dramatically different. For instance, the 5 Hz activity. In one subject, subject A is positive correlation, in subject B is negative correlation. So, which means it's difficult to generalize across subject. And that's the practical issues we want to deal with. One way to do that is to collect a lot of data, trying to look at how the human variability across different subjects, and also collect a lot of that from each individual subject. Okay, and this is a crazy study we did, and we're going to particularly look into this. So, out of range. So, the next few slides, I'm going to focus on particularly day-to-day variabilities and also the sleep quality. So we believe that the sleep quality will be one of the major factors affect your EEG and your behavior and the association between the two. So this is what we do. We ask subjects to wear this ready band, which is the wristwatch, monitor their sleep quality over 5 months straight, every single day. Oops. Can I go back? Okay. And also, each morning, they have to report their subjective feelings, including Kalinska sleepiness scale and PSD, etc. So through the cell phone. So we have those data every single day uploaded to our website. Then we can schedule subjects to come into the lab to do the experiment. So this is over 5 months, 22 weeks, and we have 22 students participate in this study. Every single day, they have to report that, and we call them up. And again, I already mentioned that they have to report subjective scores and measurement from the ready band, from the wristband, their sleep quality. So it range from 0 to 100. 100 means fully rested, and 0 means pretty much dead. And we have 3x3 repetitive measurement design. So we want to have 3 different conditions: poor sleep, good sleep, okay sleep. And each condition, we ask them to come back 3 different times within this 5 months period. So end up with Well, they are going to perform 4 hours of EEG experiment in our lab, including reaction time tests, lane keeping driving tests, I already mentioned, I already showed to you, and we collect 143 sessions over 22 weeks. Okay, so this is one of typical subjects. You can see over 5 months, and this is poor sleep, good sleep. You can see on 3 different days we record those data. Okay, and this is experiment we are subject to perform once they report to the lab. And actually, between the experiment, we also have, again, questionnaire as subject to report the subjective fatigue level and stress level, etc. Okay, so this is the behavior. When they have poor sleep, and this is their reaction time, which is higher than when they have really good sleep, which we put as a normal. And they are actually statistically significantly different. Okay, again, this is a lane keeping test. I'm going to show you the association between the EEG power spectrum and performance. Again, in this case, this is sorted according to reaction time. And when the subject has really good sleep, this is theta band, okay? And this is alpha band power. You can see that subjects that did not have really long reaction time trials. That's good, okay? And when subject has okay sleep, you can see you can extend the curve. And you can see monotonic changes of this one. And in theta band, in alpha band power, across from the short to long reaction time. And when subject has poor sleep, now you can see the differences. Particularly the alpha band power is not monotonic change. It's actually biphasic. You know, from awake to drowsiness, you can see the increase and then decrease from the deep, severe drowsiness. Okay, so this is what I show you, basically how the sleep quality will impact EEG activity and test performance. Then we're going to look at, within sessions, how the stress will impact your EEG activity. So in this case, we want to engage subject, we want to encourage subjects to do a long brain-computer interface experiment, BCI, SSEP. So we designed Candy Crush game. Okay? In this case, subjects play this game for an hour. And then they look at this, trying to search the gems and trying to see the 3 match. And then they have to guess these locations, and then we start flashing. Let me see if I can run this video. Okay, you can see we highlight 4 different areas. And one of them, if you switch 2 cells, you will make a 3 match. For instance, this one. And subject guessed the locations. Okay, you guess these locations. And this is low stress condition. Subject has 10 seconds to respond, to search. And you just fixate here. And this is stress conditions. So you have only 2.5 seconds. You have to search, send the location, basically. So subject play this for an hour. And then what we want to look at is if the EEG activity before the search, can we actually look at the signal-to-noise ratios under different conditions? In this case, you can see this is actually the 2 seconds before the search. And this is theta band power, alpha band power. This is frontal, this is occipital regions. As you can see, when the sort of fixation power or resting EEG activity power increase, they have a lower signal-to-noise ratio. And particularly when they are under stress, the slope is steeper. So which means the effect actually is more prominent, right? This is, again, this is actually 2 seconds before you see the Candy Crush. And this is the performance. This is accuracy. As you can see, again, when you're under stress, the accuracy actually decreases when you have higher resting power. So for each single trials, you can actually look at their resting EEG or their pre-stimulus EEG. It can sort of predict the signal-to-noise ratios you're going to get from the subject's EEG activities in response to the flashing stimuli. Okay, so this is to sum up. And I reported the EEG and physiological correlates of test performance of a sustained attention task in a naturalistic environment. We use realistic driving simulator. And we report EEG correlates of drowsiness can be employed to build a passive BCI to detect and mitigate drowsiness in sustained attention tasks. And also, we show the EEG correlates cognitive states vary over time within a subject and across different subjects. And we also show that real-life stress affects EEG activity, human performance, and association between the two. Last, I want to acknowledge the funding source from ONR, DARPA, Army Research Office, and ARL, and also most of Taiwan. Before I finish my talk, I want to tell you the posters and also live demo session, particular live demo session tomorrow. So that will be take place at Mobile Lab downstairs. Oh, no, actually it's over there. So for instance, this is spatial navigation experiment, and this is subject blindfolded, and then we use the motion captures in trying to look at where subject is. And this is their mental map, and this work is led by our associate director, John Iverson, and also Makoto. And this is analysis. You can see, and you can, I think Makoto is going to present a poster. And this is another study I'm working on, again, trying to look at real-life stress, how that impacts EEG activity and performance. And we developed a virtual reality escape room. And in this case, this is a top view. You can see the subjects walking into our escape room. And from time to time, we actually assess their flow, their mood, their emotions, and also we measure EEG activity, motion capture, GSR, heart rate variability, and heart rate, etc. So we are, and eye tracking as well. So we're trying to use multi-model approach to assess their response to stress. Okay. I will take a few questions.
Thanks so much.
Yes.
So I was just wondering if you. Oh, sorry. Hi. So I was wondering if you think about reaction. Let's talk about the stress, the last piece. Mm-hmm. So you say that under different stressful conditions you get different reaction times, and this is reflected in the EEG. Mm-hmm. Did you do something where you, in such a task like the game, I know you see a high stress situation versus a low stress, but if I'm playing. Time stress. High stress versus low is what
time stress, particularly. This is time stress.
Time stress.
Either 10 seconds to search the gems or 2.5 seconds.
Right, so you have these sort of two scenarios, one that's more hard than the other, right?
Yes.
But when you're playing the game, a person is actually keeping track of performance, right? So they may react differently. Like, so if they've had a streak of really easy trials, right, they might get more confident and actually do react faster if a high stress comes along.
Yes.
Versus if they've had, the opposite, right? They had a bunch of hard trials, they get an easy one and they take a long time. Yeah. I suspect that that's what might be happening here. So if you just try to say high versus low stress predicts reaction time, and therefore that's also reflected in EEG, I'm not so sure you can say that. So have you thought about, so if somebody's tracking the past, this is like the Shinachi and right, that you actually have a state variable that's accumulating evidence of the past performance and stress trials and so forth, and that's what's causing variability. And I think it's important because that's also changing your brain activity.
Yeah,
right? So
I agree with you, absolutely. And so, well, I forgot to mention, this is actually about, for maybe 10 minutes, I couldn't remember exactly the time. And they are all sort of low stress, and they're followed by the high stress conditions. So not intermittent.
Oh, so they're not interleaving?
No.
Okay.
And,
so then
but I actually studied that on tennis players. So we are subject to wear all this physiological measurement and when they are in the tennis match. So right now we're analyzing data. So for instance, when they are leading by 2 games versus they are trailing, and we're going to study that. Yeah, good question. Thanks.
Okay. Another question? No? Thank you so much for
Thank you.